If you're not on mute, please make sure you put yourselves on mute. Thank you.
Thank you. I'll get started, and thanks for the kind introduction. And for those who were on the call in the summer, this will be just a refresh. And for those who have not heard about LitVar, I hope that I'll give you a gentle introduction about what we're trying to do here. So we are, as introduced, we are a research group as part of the National Library of Medicine intramural research program, and we focus on biomedical text and image processing using machine learning and AI methods. And we spend a lot of time working on improving literature retrieval, because that's also very much tied with some of the LLM and CBI literature resources such as PubMed. But also we are very interested in creating new methods for information extraction and assisting data curation. So one of the projects that I'm going to talk about today is called LitVar. And the reason we developed LitVar is because we noticed that even though PubMed is already very powerful, but like any keyword-based searches, it is also very limited in a way that, in this example I want to show you is typically our user would search with one name of a sequence variant. And in my talk, I'm going to be using sequence variant and the mutation interchangeably, even though I know they are different. But for simplicity, I'll just using them. So as you can see, different names of the same mutation or variant would result in a very different number of articles returned. And if one user uses any one of them, so they'll get an incomplete picture of the full article set that in the literature. And so for us, can we recognize, the first question is, can we really recognize these different names of the same variation in the full text articles? And this is not a I used example, but this is a very common phenomenon in the biomedical literature. We have estimated that even though there is a nomenclature naming convention for sequence variant, but if you look at the literature, only less than 1% of the variant or mutations are written by the authors using the HGBS. In majority of the cases, they are not using following the convention. This is because I think authors are creative, also because the language is rich and ambiguous. So what we started the project, what we first did was back in 2013, we developed a tool called TMVAR, which is basically a software that would recognize different names of a variant and then normalize that into HGBS format. And then a follow-up study we called TMVAR 2.0, what we did back in 2017 was essentially to, once we have the HGBS names, and then we also find the corresponding gene for that particular variant, and then we try to normalize that using the dbSNP RSIDs. So I will show you exactly what TMVAR is capable of doing. So this is a PubMed abstract. So when you apply the TMVAR software tool, it will first extract 3 mentions of the variant, and then it will realize that these all 3 relate to the same variant. And the second step is we're using another tool called PubCurator to recognize the gene name associated with the variant in this case, and then map that to a RSID. So this is what we did using the TMVAR for normalization choice. We used RSID, and then we had a wonderful discussion at the ClinGen group meeting last time, and then several of you asked, can you normalize to a allele-specific option? So after that, we have been working with the ClinGen allele registry folks, and they were very helpful and helped us doing this. And this is still ongoing work, but we think that in addition to RSIDs, we will be able to map to CA numbers as well. And of course, we would always be able to display the normalized HGBS format as well. So these could be 3 different options of to the users how you would like to search. You can either search just by variant name, or you can search by CA number, or you can search by RSIDs. So the TMVAR tool has been evaluated on different data sets, including the data sets we created our own, and several other data sets as I showed you here. And you can see that it performs better compared to other similar tools to tag the variant in the literature. So once we developed TMVAR tool, that was a software tool that can recognize variants. And then what we did was to apply that tool, TMVAR, to the entire set of 30 million articles in PubMed and 3 million full-text articles in PMC, and then gather the tagged or tagged recognized variant and put into a database, and then display that in a searchable web front called LitVar. And this was done mainly by 2 very bright scientists in my group, Dr. Allott and Dr. Wei. And we did this first with the first version of the LitVar was launched back in 2017. And soon after, we find that LitVar is a tool that has been widely accessed by users from both academic, industry, and the different research communities, and from across the world. So because of the usage, we decided to improve, and now we are in the process of releasing the second version of the LitVar2. This is not officially launched, but we have been we consider that as a version before the pre-launch, but soft launch. But I think we have basically all the functionalities that we would like to include in this version. And what's new in LitVar2 is that it has improved search capabilities, and it has including more results as well, and the better recognition results as well. And also in terms of UI, it's also more streamlined. So in terms of search capability, in the current or the old LitVar, one can only search for a variant. But in the new LitVar2, we allow users to search both the variant as well as any other keywords, be it that keyword can be a disease name, can be a gene name, or can be a drug name, any free text word, free text. And also we have allowed and improved our search box to enable query autocomplete. This is a feature we know users are used to in the general search engine. Also, we know that from our work in working with the PubMed search, we know that PubMed users will like this as well. And also because we know that, as I alluded in the very beginning in the example, there are just many different ways a variant can be written. So when you just put in one of them, our search engine will immediately map that to many other names of the same variant, and then give you the hint which one is the one that you would like to search. So in terms of content, we have already in the previous LitVar, we have already have PubMed and PMC full-text articles processed, but previous research has shown that there are a lot of variations of sequence mutations mentioned in the supplementary materials. And so in the newer version, the 2.0 version of the LitVar, we have included processing the processed results of the supplementary materials as well. As you can see in this table, that does increase significantly the amount of papers or relevant papers or variant in the new LitVar. For example, the number of publications containing variant just in the title, abstract, and full text in the original LitVar is below 500,000, and including supplementary material will increase to 700,000. And in terms of variant and publication links, that increased from 1.8 million to 3.8 million. In addition to improving the content, to expand the content to include supplementary materials, we have also improved our TMVAR, the underlying tool for recognizing variant, and in some of the edge cases. So for example, I show you an example here that BRAF V600E in the past, such a mention would be missed by our tool because there is no space between the gene and the variant. But we've seen this kind of things over and over again. Now we have been able to process them as well. And also because we have improved our gene tagger, so we can match the variant with the corresponding gene better as well. So back to our example that I showed you earlier. So in the newer version of the LitVar2, when you search using any of those variant of the names, either using the genomic one or in the protein level or using the RSID, you will see the same number of articles returned. And in this case, that's almost 10 times higher compared to the original search if we're just using the A146T. So in terms of, we have also spent quite a amount of efforts to improve the user interface in the new LitVar. So now that we have a way to sorting papers both by relevance and by date sort order, you can also filtering on the left side is the filtering function. You can filter the search results by year and also by sections, which is quite interesting because that allows you to zoom into a certain part of the body of the text. And so if you want to find a variant only mentioned in the results, you can do that, or you want to find a variant mentioned in the discussion or in the background information introduction, you can do that as well. For each retrieved articles, we show the matching snippets and the matching publications. So if we click on one of those articles, you'll see the articles are highlighted with the variant is highlighted in red, and then there are also other entities highlighted. That's powered by our other tool called a PubTator. So the bioconcepts that we included here, including the gene, chemical, species, diseases, and the cell lines. Of course, if you can, this is an option that you can check on and check off. We also show you similar publications that related to the articles you're looking at. So LitVar has been integrated with both dbSNP and ClinVar at NCBI. So we are very pleased with that. And ultimately, and this is my last slide, but this is something that get us working on LitVar at the very beginning is we would like to extract not just the variant information alone, but the relationship between the variant and gene and diseases and unlock such information from the free text into structured data, computable knowledge. So with that, I close and thank the scientists at my lab, as well as our collaborator at dbSNP, ClinVar, Unipart, and GWAS catalog. Thank you. I look forward to the discussion.
Thank you so much, Dr. Lu. That was just a wonderful description of your work, and really looks like a great tool. So we look forward to hearing from Deb. So Deb,
sure. Hi, yeah.
Hi.
Great, thank you. I wanted to ask, can we put the link to LitVar2 in the chat? Is that, I mean, I know you mentioned it's kind of a soft launch. Are we able to share that link with the Biocurators? Okay, great. Wonderful. Okay, well, great. Okay, so now for this part, so I want to preface this by saying this is kind of my own, personal exploration through LitVar, and essentially trying to find out how can biocurators in ClinGen provide feedback for LitVar? Like what categories are needed? How can we make it a little bit more systematized so that it's not, it's just emailing a paper to you or et cetera. So we have some formalized way to get that, to send that might help to organize it a little bit more. So let me share my screen here. All right, can everyone see that?
Yep.
Great, okay.
Yes.
All right, so again, I want to say this is exploring LitVar for variant curation, doing some comparison of the variants to a manually annotated set that we have that focuses on the VHL literature that is in Hypothesis, which is an annotation program, a literature annotation program. And then understanding the ways that we can perhaps use hypothesis, or that if we are finding papers, even separate from using hypothesis, how can we give that feedback to LitVar? And then what type would be best to be most useful. So here I've put together, we did have the LitVar call on the consortium call. We have a wonderful presentation by Jessica Mester on finding literature for variant curation. And then as well, the gene curation SOP has a great section on literature searching techniques and tools as well. This is just kind of a summary of some of the guidance that we've had recently for it. So my goal here is to compare LitVar2 searches to a literature set that was compiled by the VHL team using hypothesis. And then also to answer some questions, do they find the same articles? Why or why not? Do I get useful articles with LitVar2? Why or why not? And then some cases of missing. So I think some of the cases that I've been most interested in is when LitVar2 misses an article that's found by the hypothesis team. So this means a human has manually reviewed this paper and has said, this is an important paper, but LitVar2 doesn't find it. So those are really the cases that I'm the most interested in, and not necessarily, comparing the numbers of cases of what's found, but really these edge cases maybe, or unknowns as to why it wasn't found. So I want to just give a little background on the VHL hypothesis set, and as well acknowledge all of the work that has gone into it through the ClinGen C3, as well as members of Raymond Kim's group at UHN Toronto. So this is a set that was developed through a student curator process, where they developed a protocol to do annotation on this literature. They did a deep literature search to obtain the literature on the VHL gene and variants. And then they've created these specific annotations, and they've done it in conversation with the variant curation expert panel. And then over time now, they have trained new curators and are adding to that literature set. And so I just want to show quickly, this is, for instance, what a search might look like. So now in the VHL VSEP, we go to our hypothesis set, we do a search on a variant, and then pull up some papers, and then further, we can pull up the annotations on those papers. And this is an example just of what some of the annotations might look like as well. And this is an overview here of how we've been using it so far. The hypothesis team created this literature set. They tell us that the curation is complete on a shared worksheet. So they list the number of papers that they found, and then the papers that they've annotated. We then go through it with our VSEP and with the pilot variants that we intend to curate. We discuss it with the team because there could be some oddities in the literature about that variant. So there's sometimes conflations or redundancy in the literature. Then we prioritize based on that, and we use that as one of the inputs, along with other inputs for the VSEP, then to do the curation. So just a quick note here that I think it's a good idea to put together a literature curation group, and using hypothesis is a good way to do that. But to do that in advance, while the evidence code specification step is going on, and then that way that set can be prepared. So that's just a little bit of background on this set and on our use of it. So again, this is the goals to just basically answer some questions on looking at LitVar2. So here is the list that we have of the variants, and here is the papers that the hypothesis team has put together. And again, we do a lot of prioritization based on their completeness. They found a certain number of papers, then they show that they've annotated them all, and they make some notes about it as well. So what I did here is I didn't start with the ones that have the most papers, but I started with ones that kind of have, a moderate number. So one of the first ones that I looked at is Pro 25 Lu here. And just went through these papers. So I ran the search within LitVar, and then I looked in the papers that we have in the hypothesis set as well. And I want to say some caveats here. Number one is the search string. So actually in hypothesis, the search string of the variant itself is not the most ideal search. It's also using the ClinVar ID, which I did not do here. And second is that there is some uniqueness to how the set is put together in hypothesis, or some choices that are made. And some of those are if a paper is very highly redundant or contains samples that we just could not feasibly use, then they don't put it into the set. So there is some, again, the intent is not really to compare the numbers in between it, but mostly to look at these cases where when we get articles back from literature search engines, they're either just totally off, or cases where interesting articles have been found through manual curation that aren't found through the literature curation. And so I think we all experience that in doing literature searches and in using tools for literature searches. So compiling some of this together and figuring out reasons why is what I'm interested in. So here, just generally, these are the total articles that we had in hypothesis at the time. This was the articles that we found with LitVar2. There was some overlap of articles, and actually there's more overlap most likely because I missed some articles in the hypothesis set. But here's where I put together a table of, this is the articles that are unique to LitVar2. And again, for instance, this one is actually in the hypothesis set after all, but those aren't really the ones I'm interested in. So here would, for example, be a spurious match. So this is where, and I put together some labels, which would, like, one label would be more descriptive, technically correct, practically wrong. So the gene variant had like the variant in the gene had a mislocation. Interestingly, something like these articles would not even make it into our kind of smaller set because they would be excluded as they're off target for the gene, for the disease in the first place. Another issue, particularly for cancer, is that we have this issue of purely somatic papers. So in this case, we're looking for inherited variants. And if a paper is only about the tumor samples, then we also might not be interested in that paper for this case. Another issue is, technically correct, but a very brief reference or very redundant. So it's like, as a curator, we don't need all of the papers ever about that, but we need like the important papers about that topic, or we need the most relevant papers. So again, this is my thoughts of some tags here, and then ultimately tagging them kind of in a binary way, like useful, not useful, for that. So I just want to give a couple examples here of one of these. So this is where it's this mitochondrial inherited variant, and so this is technically correct because you have what I searched for, P25L, and then also the VHL gene, but the context is that P25L is mapping to this ND6 gene. So it's actually P25 in the ND6 gene. And so considering of some spatial relation in between the two genes, right? So trying to standardize that when we give that feedback back to LitVar would be great. And if it's just saying, technically correct and practically wrong or something, I don't know exactly how to phrase it or how that would be most helpful. I want to do a couple of other examples, and then we can open it up for some discussion as well. So in this case here, the topic has a different disease than the disease of interest. And so it is this, they do mention this variant. So this is, again, technically correct, but it's not useful for the curation. And so ways to exclude that, I guess, would be also including the disease, but in this case, the disease is also the gene's actual name. So that's quite difficult to exclude in any way. And so then there were also 10, let's see, I skipped this here. There were 10 articles that were unique to the hypothesis set. And again, these are the ones I'm most interested in. And so labels for, for why a manual set would have found it that LitVar did not find it. And I know that one of the issues is access. And so several of them say, I can see that the article is not freely available. So it's not in PubMed Central, et cetera. But others say free access. But then I recall on the consortium call you mentioned, they still have restrictions about computational use. So that's very difficult to kind of tease apart here because, there's no obvious reason I can see, plus the article says free, but it's not free for this use. So I tried to kind of take a guess at some of that. But here, for instance, this is one that I identified of interest, which is this spelling issue as well. So in the entire article, they don't put the variant in any format that's really recognizable. They have proline 25-2 leucine. And so again, this would be something I'd like to send back to LitVar, just so it's on the radar as well as another way that this is spelled in the literature. So I think that, I have a couple of other ones from here. And again, if I couldn't figure out a reason for it, then I just put unknown here. So if it's access, but otherwise, if it said free and I couldn't figure out why it wasn't found, then I labeled it as unknown. So these were a couple of other just, kind of questions in the use of LitVar, but I want to pause here before we go on to these ones, to just talk about the feedback mechanisms and what ways would be useful.
Well, thank you.
Yeah.
This is very interesting, and I think will be very useful for us. But let me understand this a little bit. So, how does the hypothesis team find the set of papers? Do you know?
Yeah. So they worked with and Courtney, if is on the call, could speak to this too. This team worked with a librarian at I think part of Raymond Kim's group, or through Raymond Kim's group, and had them do a professional search, I guess, or a deep literature search on it. I'm assuming it used PubMed, but I don't know the exact criteria that was used for it to get this literature set in the first place. And I know that it has been added to over time, but they did stop that search. I think up to 2019 or something. That's when they cut it off, and have not done that type of search again.
So this was completely a manual search, meaning they put keywords. Maybe put a bunch of different keywords in PubMed or any other search engine, and then look at the papers, and they come up with a set of papers they deem relevant for curation purposes.
Yes, that's my sense of it, and that they also you kind of seed your search by reading one paper. That paper has good references. You get those other references. So I'm sure that it was keyword search plus expertise in the literature informing it, and then over time finding the good references from reading the initial papers. Right,
because the current way that LitVar set up, and we trying to return all the papers containing that variant, but we're not limiting or doing any filtering to say these are the papers that is most likely to be useful for ClinGen group or for another group. Okay, So that's something I think is something that appealing to the curators, because you do not want to read all the papers. You want to read the important papers, or the papers that are so that's one thing. That's certainly something that we can try to do, but we would need some examples like the hypothesis group has done, for this variant, and they have showed that these papers are more relevant. Or we have other papers that you have curated in the past, given a variant, these set of papers are relevant, but other papers are not so relevant, even though they contain that variant. So those are the examples that we would like to consider, and then trying to learn, differentiate between the papers, even though, let's say, given a variant and the disease name, there are 100 papers returned, can be found in LitVar, but only 20 were curated. And
why? Yeah.
Why is that? And especially the other 80 has been humanly reviewed and decided not to curate or not to including as evidence. So those are the things that we definitely appreciate. So that's one thing. And the other thing is, as you alluded, yes, we have a issue of access. So in the PubMed Central database, there are more papers that are free to read, but not necessarily we have the copyright to text and data mine. Okay,
Right.
We have a subset of the PMC articles that allow us to do text and data mining, and that's what we include or in LitVar2. But there are still a portion of the articles that we do not have copyright. So even though you can free access, you can search that and you can read that paper, but we just cannot process them, and that will not be included here. So that I would think accounts for several examples here.
Right,
and then,
access.
Yeah, and the third comment is, yes, there are edge cases like what that one example, they describe the variant completely in natural language. We have some ways to do that, but this is certainly something I guess we missed probably in this case.
But adding, I wanted to ask, so adding this in, so structuring some query or trying to find this in the paper, then how much does that open it up to false hits, like hits that?
Yeah, so that's what we always do, is we do not want to work on just one example, because one example can also lead to false hits of others.
Right.
But glad what we do is we have a set of sort of a benchmarking data set that we can always evaluate any change to our system that would only including new things, incorporate new things that would help overall performance, but not necessarily one case by one case.
Yeah. Okay.
That's our general rule of improving the tagger. And then my last question is, did you find papers in LitVar2 which is not in the there are a few papers not in the hypothesis, but are they useful? The ones that only
right. So that I would need to go. So certainly the supplementary ones is the ones, and I only have one example here. There are other examples. So in this case, there's a couple of reasons that's a hard question to answer. And even if the search string, so I searched using the variant, but actually searching using the ClinVar ID is useful, but then not all of them have a ClinVar ID. So in terms of, the searching aspect of hypothesis is one reason why this might not be as complete. But another is that LitVar would find useful papers that the manual set would not find, because at the time the manual set is put together, they're really focusing on case data and they're looking for cases. Whereas, for instance, something like this, which again, I don't know if I would need to go back and check, but this is has a variant in the gene, but it's about tumor data, but it has functional data in it. And so the functional data is also something we're interested in and we can use. We don't have to have just case data. So there are several reasons as to why it wouldn't be in the in the hypothesis set. And those are kind of decision making by the people who put the literature together. So yeah, I guess I'm just trying to find out, and I'd love for other biocurators as well who have either used this or have interest in this. Again, we can find edge cases all the time. How to put those together? When does like, if I start seeing this all the time, like 5 instances, then do we send it to you? How can we kind of structure it and organize it? Or for instance, with the you said about the paper set that we don't use, getting together a literature set that we discard.
Yeah,
you know,
that's very useful set. That would be useful set, really.
Okay.
And the other question I have is, and to the biocurators here on the call, so how do you do a literature search right now? I sort of a little bit lost here. Is that are you trying to using LitVar as a step after you have manual collection of the papers through the hypothesis tool, or are you trying to using LitVar as another like search engine like PubMed or Google Scholar, just searching for your literature at the very beginning? Where is literature search? Where is literature search in the process of your curation?
I would love for some other biocurators to speak up, so it's not just like my personal workflow.
So this is Jenny. I can tell you what I do for our GAA variants, and we do have a hypothesis set, but it's not a complete set. So I actually typically start with the free version of Mastermind, which is free, which is an advantage, but as many of you may know, it does limit the number of searches that you can do. And then typically we'll go to Google Scholar and then also do a LitVar search. I use a bunch of different tools because I feel like different searches sometimes return additional papers. So those are the main things that I use. There's also actually, luckily for Pompe disease and GAA, there's a really nice locus-specific database. So I use all of these different tools in order to find the papers that I curate. And just, as I'm listening to this discussion, I feel like, one of the, I guess, key issues is the balance between returning as many articles as possible and the sort of specificity of those articles. I do find with Mastermind as well sometimes that it will return articles that maybe aren't even for the gene that I'm interested in. It just seems to be kind of random. So, but I would almost rather have more things that I can then sort through than be missing key pieces of information. So I guess, specificity is hard, but I don't want to be missing things. I'd rather kind of kick out things that I don't need than miss an important article, if that helps.
So you also mentioned that you also, for your curation project, you also have a set of articles already manually collected by the hypothesis team. Is that correct?
Yes, and
is that sort of a general practice for all the curators at ClinGen? You always, for the genes that you curate, you have a set of seed papers collected manually by the hypothesis team to start with.
Well,
yes.
The lucky ones.
Yeah, and I think it's kind of gene dependent, and one of the issues, I guess, is just, obviously it takes time to annotate those papers. Again, for GAA, we identified those via a PubMed search and have student volunteers who are going through them and annotating. But because there are so many papers, we just don't have a full set. But I know for some genes, they do. I think the platelet disorders has a pretty full set for their genes, because it takes time for the curators to go through and annotate everything. So it certainly is like super helpful as a tool. But as you alluded to, Dr. Lu, how were those articles first identified? And I think it sounds really nice what the VHL team did in working with a librarian. I would say for other genes, it's been very much using sort of a PubMed search or other, like maybe Google Scholar and things like that. I don't think everyone has had a librarian that they could work with to identify these articles.
Right. I was just thinking that during the manual collection by the hypothesis team their manual collection, they could also using LitVar as part of the tools.
Right. So in this case, I mean, so for VHL, I don't think they did because that search was cut off at 2019. So I don't think they did. But I guess too, you know, part of this came in terms of I struggle with like relevancy of papers. So there is specificity and, you know, finding the exact thing, but again, it's finding like not just the match, but the usefulness, the relevance. So this came a little bit because of redundancy in the VHL literature. So VHL, maybe because it's an older gene, they have papers that, I mean, they will publish on the same cases over and over and over. And so reading through those only to find out that, oh, it's actually the same case is a big time sink, right? So it was really in looking too at redundant papers and this question of pulling out redundancy. So we have a set of literature that we've kind of set aside in the VHL space where it's just replicative articles, frankly, like it doesn't add much. So then giving that set to LitVar or having LitVar have that as a redundant article set, right? So again, I don't know, is that, how do we even send it to you? Do you just want to send papers or PMID, like, you know?
Yeah, well, you don't, yeah, no, those kind of papers examples will be very useful so that we can analyze the papers and understand, you know, what basically what we're trying to do is trying to learn from humans. You know, if you know these a set of redundant papers, can we try to learn, like to teach computers to learn, and these are redundant papers, and if you can learn well, then the next time when we have redundant papers, we can automatically flag that as redundant. So you don't need to send us in cases like examples, you don't need to send us the full paper, you can just send a PMID, that would be sufficient. Well, we would be able to pull the article's text conveniently here locally. Okay. So yeah, so let me just ask one. The other question I have is, so when you search, are you typically just search for using the, you know, what are typical searches you perform in this case? Not just LitVar, but, you know, Mastermind or PubMed or Google Scholar. Is this just you search for the variant and the gene and the disease? Is that the three things, like triplets, that you typically, you know, that's you call that a complete set of things that you want to find in the paper? Or most likely, you know, if you don't have the disease, you just have the gene and the variant, you do that as well? Or just the gene and the disease? You know, tell us a little bit about, you know, what typical information needs or search terms.
You know, I think that it's all over the place, and I would love to start keeping track of search strings. Like what are the search strings that we do, and to have those in a file, like even a Google sheet, we could just, if you use a search string, put it here, and then understand what are the characteristics of searching. So I know in my case, it's sometimes dependent upon on the gene name. So for instance, POL-E is very difficult to search for because it's also the word pole. And so you have to use a lot of other search strings and a lot of other things to actually pull out relevant literature about the gene POL-E. But other gene names might be unique enough that then you don't need to use such specific search strings for it.
Sorry for interrupting. So do you say that in your searches you can search just a gene, but not related to the variant, or you always?
Yeah, Sometimes, so if we're doing gene curation, and then we might just want to pull up articles that are not necessarily dependent upon a variant, but they would have expression information or some other type of information. And so if that gene name is unique enough, you might be able to just search the gene name and pull up some literature from it to curate on.
Yeah. So just be mindful that for LitVar, if we just search the gene, you can do that, I believe, but it would only return the subset of articles that also contains at least one variant.
Okay.
So if a paper is just about that gene, but there's no mutations mentioned, then LitVar is not going to return you that paper. And that, that would be some other tools that we have developed for this purpose. But
okay, that's good to know. Yeah.
So if that's a common theme of ClinGen curators, then we might adding that gene only papers, not, in other words, not necessarily regardless the presence or absence of variant, we would always return you want that paper. We can also do that in the LitVar future versions. But just we let you know that right now, if we just search for the gene without the variant, you likely miss some papers.
Yeah. Okay, that's good to know. I didn't know that. I mean, I haven't used LitVar2 to search just for gene because it variant is in the name. So I have a variant to use it. But
and the other thing I want to ask all the curators or that you have now using, are there the features that you particularly like in PubMed, Google Scholar, or Mastermind that you just find that this is, if I were to search, this is something that I really like from those platforms or search engines, so that we can see whether that feature is in LitVar or not in LitVar, and if it's not in LitVar, can we think of doing something similar?
I'd love to hear from other biocurators on it too. Anyone is don't be shy. And in the chat too, I think we are, oh great. So there are some questions here in the chat. Does anyone have a response here? There we go. We have a raised hand here. Jer? You might be on mute or we are getting a couple responses here. So one is the filter out cancer related articles. I always want to filter those in. But I suppose for groups that might be
related to the comment you made earlier regarding semantic versus
yeah, probably. Yeah. So there are diseases where the gene may be the cause of somatic tumors, but there's an underlying hereditary condition that is unrelated to the tumors. And so then if you're trying to search the literature for that, you just have an overwhelming number sometimes of tumor specific cancer papers, which are not the meat of what you're looking for. They could contain useful information, but not the most relevant. Another question here is, can we have both stringent and a relaxed filter functions? So I guess filtering on the set that you get returned and being able to have some filtering features.
Well, I'm not quite sure about understand this question, stringent and relaxed filter function.
This is Sholin. I can explain a little bit more. I think that when I using Google Scholar and try to find the gene and the variants, and that is sometimes come with either the gene or the variants. But sometimes we need both. In that way, I think that if you have the option of being stringent, exactly text match, and the option of relaxing, basically match one of them, so that you have a tiered search strategy in terms of the information you want to get.
I see. So when you want to relax, do you mean that papers when they do
Like
not
match
only contain
any term, match any one term, two term, or something like that. That kind of option.
Yeah. That's certainly something possible to be implemented, I think.
Thank you.
And we have one more here from Joanna. So there's a few questions in there. One says, why does the variant on the page appear only at the protein level and not the DNA level? I think that it does. Oh, not at like the genomic coordinates. Is that?
We do find papers on the DNA level as well. As well. So, and in fact, if you search both using the DNA genomic ACTG versus the protein level, you should return. You should see the same amount of papers returned.
Right. So here, for instance, here's P. If I put in P25L and I'm looking for VHL, so I can put P25L, then it shows here. I guess in this case, it doesn't show the C dot level. That might be.
It does not show the C dot level, but it's the results. If there is a papers using the C dot, it will be returned. Okay, so in other words,
yeah.
Right,
okay.
Go back to the home page. Oh,
yeah.
see here. There's
okay,
example.
sorry. There.
Here's an example here.
Okay, yes. So this highlights here as well.
Yes, maybe I can clarify my question.
Sure.
My question is, if there's two variants that turn the protein P25L, how do you distinguish between them? So if you have C.74C to T and C.74C to A, for example, and both of them turn the same protein change, how do you
different
distinguish
nucleotide
them?
changes? Yes. Okay. Different nucleotide changes that cause the same protein change. Is that distinguished in LitVar or not?
Right now, I think I think if they are, well, depending on your search terms, if you search for the specific genomic DNA level, it's going to be different. But if you search for the protein level, I think the results are the same because we map to the RSID, which does not really differentiate between the alleles. But like I said, we are working with the ClinGen allele registry so that it will be allele specific.
Oh, great. Okay.
In that case, it will be two separate variants when you search them.
Okay, thank you.
And it's Jenny. I just wanted to point out too that I think it's always challenging to figure out what your search strings are, especially for something like Google Scholar. And I know that Jesse Mester gave that really nice talk on literature searches before. The thing with hypothesis is that these annotators are going through and actually figuring out what the ClinVar ID or CA ID is for these variants. So it does give you a very specific search, once that annotation has been done. But I guess, first identifying that literature is kind of what we're sort of focusing on here. So hypothesis is like another step beyond that, where the annotators have gone through, figured out the IDs, and really picked out the important information as well. But I just wanted to make sure that everyone on the call is kind of understands the difference and also really the advantages of the hypothesis annotation, and also the great work that's being done here in terms of trying to figure out what are the best search mechanisms in order to identify that literature.
Like I mentioned, I think once we have incorporated the CA numbers, then the users would be able to, if you have already figured out the CA number for your variant, you can just put the CA numbers and all the papers, either they mention the protein level or the DNA level, they'll all be returned.
That's very helpful.
Right now, you can do that with the RSIDs, but not the CA. But that's something we're working on right now.
That's fantastic.
That's
really great.
That's a feedback, from the last ClinGen discussion.
That's great.
So
it's going to be really helpful.
For cases like feedback, we really would appreciate the feedback because I think that would help us to improve. And also, the more feedback you can provide, and it's actually something that we can make the LitVar be more relevant for your creation purposes. In other words, the papers we can still find the same set of papers, but we can rank them so that the most relevant ones would be the ones that to your curation purposes. And that's what we can do. But we have to learn from the examples that you have curated. And that's something I think we would appreciate examples from you. And especially the ones that you have curated versus the ones you have reviewed but rejected as well. So those are important set of papers as well. In terms of individual cases where you see that the papers are just that the variant was not tagged correctly, and there are cases like that, even though we have over 90% of accuracy, but it's never going to be perfect. It's not perfect, and it's probably never going to be perfect. But it doesn't mean that they're not useful for you. But we would appreciate. I don't know what the best way to do this, but we can certainly set up like a Google spreadsheet or something that it can be shared with the ClinGen curators, and you can just, if it's a ad hoc, you just search something and you don't find that relevant or useful, and then you just put on that spreadsheet and put on programmatic for you, and then we will be able to look at, or you can send us emails. But either way, we would appreciate your feedback.
Great. Well, all right, we're at the top of the hour. So Jenny, did you have any follow-up slides or anything here? Otherwise, I want to really thank the presenter, Dr. Lu, and the group as well. And I think that, I would love to continue working on this. And so if there are other biocurators interested to kind of get some more formalization to this feedback, or to just regularly provide feedback, that would be great.
Yeah, that sounds great. And thank you so much. Thank you, Dr. Lu, for all your great work, and Deb for looking into this as well. This has been really helpful. So thank you very much to you both.
All right, thank you. Bye.
Thanks so much. Take care.